السلام عليكم جماعات. أفريقيا موضوعنا من برنامج structure of proteins, tertiary and quaternary structures of proteins. The overall three-dimensional arrangement of all atoms in a protein is referred to as the protein tertiary structure. Whereas يعني tertiary structure يعني انت بلنداهي هيا فلاي هيا طول وعرض وارتفاع تاعها بلنداهي بلنداهي تاعها بحنكي ودرجة هي. Whereas the term secondary structure refers to the spatial arrangement of amino acid residues that are adjacent in the primary structure. Tertiary structure includes longer range aspects of amino acid sequence. Amino acids that are far apart in the polypeptide sequence and that reside in different types of secondary structure may interact within the completely folded structure of protein. The location of bands, including turns in the polypeptide chains, and the direction and angle of these bands are determined by the number and location of specific band producing residues such as proline, threonine, serine, and glycine. باش الرسم إن شاء الله هم بيشرحين. Interacting segments of polypeptide chains are held in their characteristic tertiary position by different kinds of weak bonding interaction and sometimes by covalent bonds such as disulfide cross-link between the segments. Some proteins contain two or more separate polypeptide chains or subunits which may be identical or different. The arrangement of these protein subunits in three-dimensional complexes constitutes quaternary structure. In considering this higher level of structure, it is useful to classify protein into two major groups. قال جورا أمشين بروتينا دابش بكن السر الأساسي structurally fibrous proteins globular protein. Fibrous having polypeptide chains arranged in long strands or sheets. Fibrous وك حلقانة وك شيتا وك ورقانة شد. But globular proteins having polypeptide chains folded into a spherical or globular shape بشكل very grover نادرش كانيا وك ورقيا. The two groups are structurally distinct and have هردو جوازن. A fibrous protein usually consists largely of a single type of secondary structure globular يعني above fibrous on helixa on sheeta. Globular protein often contains several types of secondary structure. نقال جا helix أو sheeta هردو بيكفنا. هبجوازين مهمة بين globular أو fibrous protein. The two groups differ functionally in that هردو structure هردو كجك شولي هردو كجك قال جواز شيء. In that the structure that provides support, shape, and external protection together are made of fibrous protein. Fibrous protein بهرابة شولي في structure دتا خانة و extracellular و organelles وأكتشتها وبس بهرابة اش انزيما globular protein. Protein quaternary structure range from simple diamonds to large complexes. Quaternary يعني شنو في ديارة دا چار polypeptide هنا بيجوا bit X structure. Protein architecture-quaternary structure. Many proteins have multiple polypeptide subunits. يعني أكو إك بروتينة بس چار فرع هنا. Many multisubunit proteins have regulatory roles. Like enzyme, the binding of small molecule may affect the interaction between subunits, causing large change in the protein activity in response to small change in the concentration of substrate or regulatory molecules. In other cases, separate subunits can take on separate but related functions such as catalysis and regulation. Some associations such as the fibrous protein and the coat proteins of viruses serve primarily structural roles. Some very large protein assemblies are the size of complex multi-step reaction. One example is the ribosome. Protein synthesis. A ribosome is the site of protein synthesis, which incorporates dozens of protein subunits along with a number of RNA molecules. A multisubunit protein is also referred to as the multimer. Multimeric protein can have from 2 to hundreds of subunits. A multimer with just a few subunits is called an oligomer. If a multimer is composed of a number of non-identical subunits, the overall structure of the protein can be asymmetric and quite complicated. Although most multimers have identical subunits or repeating groups of non-identical subunits, usually in asymmetric arrangements. The first oligomeric protein for which the three-dimensional structure was determined was hemoglobin. هناك oligomeric protein يعني quaternary structure hemoglobin. Avohane epicotiesh 4 polypeptide chains and 4 heme prosthetic group in which the iron atoms are in the ferrous state, Fe+2. The protein portion called globin, hemoglobin, globin protein, heme non-protein part. Hemoglobin consists of 2 alpha chains and 2 beta chains. يعني دوشوان alpha ودوشوان beta. أبا structure hemoglobini أبا structure أبا هيما وأبا هيموغلوبين. أبا هني هيما وأبا globina. أبا هني structure همي بجت سلسلة أبا نبتش globina. أبا هيما وأبا وصا أبا هني أبا مازن كذا iron النيفيا دانا 4 أبا 1 أبا 2 أبا 3 أبا 4 4 prosthetic group heme 4 heme أبا هاد 4 heme وأكسجين الفيريفا أو صار أبا جهة جفالة كو أكسجين شيت أوربيتال أخوات دينت تيدا وتفاعل بكتنو لو ما هيموغلوبين شيت أكسجيني نقل كتن. Because hemoglobin is 4 times as large as myoglobin, much more time and effort were required to solve its three-dimensional structure by X-ray analysis, finally achieved by Max Perottis, John Kindros, and their colleagues in 1959. The subunits of hemoglobin are arranged in symmetric pairs, each pair having 1 alpha and 1 beta subunits. Loss of protein structure can get protein structure جداسد شو لخوا داسد دم protein structure خوا داسد اف چند هنی وقت کتن کو شو لخوا شکش داس بدتن يعني هكا تو جام كي بروتيني on pH زيادة كي on كيم كي هذا هميدي وقت structure بدل بتو بالمقابل شو لي بيش بدل بتو. Protein structure have evolved to function in particular cellular environments. The conditions different from those in the cell can result in protein structural changes large and small. A loss of three-dimensional structure sufficient to cause loss of function is called denaturation. Denaturation is a loss of three-dimensional structure of a protein leading to loss of function. The denatured state doesn't necessarily equate with complete unfolding the protein and randomization of conformation. Under most conditions, denatured protein exists in a set of partially folded states that are poorly understood. Most protein can be denatured by heat, which affects the weak interaction in a protein, primarily hydrogen bonds, in a complex manner. If the temperature is increased slowly, a protein conformation generally remains intact until an abrupt loss of structure occurs over a narrow temperature range. The abruptness of the change suggests that unfolding is a cooperative process. Loss of structure is when part of the protein destabilizes other parts. Protein can be denatured not only by heat. Gangrene یک کنه که denaturation of protein چه also denaturation can also happen through pH. Changing the pH will change the structure of protein by certain miscible organic solvents such as alcohol or acetone, by certain solutes such as urea, guanidine, hydrochloride, or by detergent. All these solutions can change pH of the protein, leading to change in the structure and ultimately change in function or loss of functions. Each of these denaturing agents represent a relatively mild treatment in the sense that no covalent bonds in the polypeptide chains are broken. Organic solvent, urea, and detergents act primarily by disrupting the hydrophobic interaction that make up the stable core of globular protein. Stream of pH alter the net charge of the protein, causing electrostatic repulsion and the disruption of some hydrogen bonding. This is the mechanism of how pH can change the structure of the protein. pH basically disrupt the hydrophobic interaction between the globular protein, between the amino acid, causing repulsion. For example, if pH is acidic, there will be more positive charge on the protein, so this will lead to repulsion. If it's basic, more OH, more negative charge, and again more repulsion and changing the structure, changing the three-dimensional structure of the protein, and then loss of function. The denatured states obtained with these various treatments need not to be equivalent. Oxygen can be bound to a heme prosthetic group. As we said, oxygen is poorly soluble aqueous solution and cannot be carried out to tissues in sufficient quantity. It is simply dissolved in blood serum. Dissolved oxygen is poorly soluble in serum or in water. That's why it's needed. It needs a carrier to be transported, and the carrier is hemoglobin. Diffusion of oxygen through tissue is also ineffective over distances greater than a few millimeters. We have a phenomenon called diffusion, transferring of molecule from high concentration to low concentration, and this kind of phenomenon is poor for oxygen to transfer. So the ideal transporter is hemoglobin. The evolution of larger multicellular will depend on the evolution of protein that could transport and store oxygen. However, none of the amino acid side chains in protein is suited for the reversible binding of oxygen molecules. This role is filled by certain transition metals, among them iron and copper, that have strong tendency to bind oxygen. Multicellular organisms exploit the properties of metals, most commonly iron, for oxygen transport. However, free iron promotes the formation of highly reactive oxygen species such as hydroxyl radical that can damage DNA and other macromolecules. Iron هكذا ده بلا شماتها جكاروسا خرابة إجبار iron تشيتن عندك molecular شيكتن نوفونا كو radical سروانا او راديكال هني جلا اكتيفان شيت تفاعلي قال دي ان اي بكت ودي ان اي بيش بكت شيت تفاعلي قال انزيم بروتين بكت وبروتين بيش بكت وهرسل. Iron use in cell is therefore bound in form that sequester it and or make it less reactive. In multicellular organisms, especially those in which iron and its oxygen carrying capacity must be transported over large distances, iron is often incorporated into a protein-bound prosthetic group called heme. Heme consists of a complex organic ring structure, protoporphyrin, to which is bound a single iron atom in its ferrous state. The iron atom has 6 coordination bonds, 4 to nitrogen atoms that are part of the flat porphyrin ring system and 2 perpendicular to the porphyrin. Heme is found in a number of oxygen transporting proteins as well as in some proteins such as the cytochrome that participate in oxidation reduction in electron transport chain reactions. One of these two coordination bond is occupied by a single chain nitrogen of acetylene residue. The other is the binding site for molecular oxygen. This is the structure of hemoglobin. Heme. When oxygen binds, the electronic properties of heme iron change. This accounts for the change in color from the dark purple of oxygen-depleted venous blood sample to the bright. قال جرى كيف فرقنا بين شرياني وريدي هيا دمه شريانك تسحكيك وريد شينا dark purple بي بازياكيا of oxygen depleted اگر oxygen كم بتن بس هكا oxygen زيادة بت رنجي دي الصور لقيتن لو ما يا شريانا رنجي برايت ريدا رنجي صورة بس أذي وقت بي بازيانا وقت أوانا أي وريدية oxygen وريكيما. Some small molecules such as carbon monoxide and nitric oxide coordinate to heme iron with greater affinity than does. قالك عندك مولكيول هنا جهراوينا وك كربون مونوكسيد على نيتريك اوكسيد بوتي جهراوينا شكو الشيت تفاعلي قال الهيموغلوبيني بكت وتفاعلي بهيس كوكت الهيموغلوبيني نفبت هذا هيموغلوبين دي كيمبت وبالمقابل دي بتا قال مرنج اوكسجينات تونايت. When a molecule of carbon monoxide is bound to heme, O2 is excluded, which is why carbon monoxide is highly toxic to aerobic organisms. Oxygen is transported in a blood by hemoglobin. Nearly all the oxygen carried by a whole blood in animal is bound and transported by hemoglobin in erythrocyte, red blood cells. Normal human erythrocytes are small, 6 to 9 millimeter in diameter, bioconcave. They are formed from precursor stem cells called a hemocytoblast. روش خانكي بچيكا هذا بلاصات الريثروسايت بايكونكيف يعني قرصاء محقرة هردو all over إنه كان مشكلة دائما. They are formed from precursor stem cells called hemocytoblast. In the maturation process, the stem cell produces daughter cells that form large amount of hemoglobin and then lose their intracellular nucleus, mitochondria, nucleus, whenever أوليكا hemocytoblast دي بتشيكا هيش نيخودها شتينجي دي معظم بت وقتی معظم بتش عملية ميتوكوندريا خوش دست دت اندوبلازم ودي مش دست شو دست دت والبتا شي البتا إريثروسايت هي red blood cells. erythrocytes are thus incomplete يعني نكاملة خانك نكاملة ما تكون dilated anemia unable to produce and in humans destined to survive for only about 120 days. وعاملة يجي تقريبا 6 شهور هيبقى كان red blood cell تبدأ تملت. Their main function is to carry hemoglobin of red blood cell شو اللي بتشيا جالك hemoglobin هالت قرتك وشو اللي هي أكسجيني نقلت كده which is dissolved the cytosol at a very high concentration. In arterial blood passing from the lungs through the heart to the peripheral tissues, hemoglobin is about 96 saturated with oxygen. وتوشش صادي شوان هيموغلوبينا أفيت سهيدا هم بيبت أكسجين وشنيكا دات كابتن شويري بلا رابط بو دماغي وبو كلية وبو ميناكي وبو همي جهة دا أكسجين يمدتين. In the venous blood returning to the heart أبا الزفرتين هيموغلوبينا الزفرت الشهاش دماغي وبو جسكي الزفرتين كيما أو هيموغلوبينا أكسجين تدا كيما تقريبا 64 صادي أكسجين بيفا كيمياء دزفر السهيبة دابوخا أكسجيني بترورجتنو بينسبة بالعراكتن. Thus, each 100 ml of blood passing through a tissue releases about 1/3 of the oxygen it carries, or 6.5 ml of oxygen gas at atmospheric pressure and body temperature. Sickle cell anemia. أبا بلا مين محاضرة